Hello, Mr. P here. Bulk transport is the purpose of lecture 9. And so in this lecture, it's going to be a really short lecture. At least I've promised all my classes that it will be. And so we're going to try to keep this to 5 minutes or less. So bulk transport is a form of active transport. In the last lecture, we talked about the difference between passive and active transport. We know that passive transport requires no energy, and active transport does require energy. Active transport also requires a membrane and specific protein channels or pumps, where passive transport can use a membrane and can use a protein, but it doesn't necessarily need one. And so when we talk about bulk transport, remember that we're talking about a form of active transport, and we're using membranes that fold around particles that are eventually going to take them places. And so bulk transport is made up of two main processes. One is endocytosis and one is exocytosis. Endocytosis is what it sounds like. It's taking materials into a cell. Endo means into, and exo means out of or exiting. And so endocytosis is taking materials in, and we're talking about large materials. And exocytosis is taking materials out of the cell or excreting materials out of the cell. Cells will excrete proteins or enzymes, and cells will bring in specific proteins and enzymes from other neighboring cells or other tissue cells. Okay, so cells are always talking to each other, they're always communicating, and one of the ways in which they communicate and in which they do certain cellular processes is through the process of endo and exocytosis. Endocytosis specifically is about how cells take materials into the cytoplasm or take them in from outside of the cell. And so it's a form of active transport for large materials, and there's two different types of endocytosis. One is phagocytosis and the other is penocytosis, and we'll talk a little bit about both of those. But remember, endocytosis is bringing materials into the cell, and again, it's a form of active transport, so it requires energy. Here in A, this is a form of endocytosis. See, we've got materials that may be food molecules, it may be bacteria, it may be other types of proteins or something like that. And so these will come in, they'll pinch this membrane in, and this membrane will eventually enclose them. And eventually we have an entire vesicle that will be a transport vesicle, and that vesicle will transport these materials in this membrane-bound bubble all the way through the cell, and so they'll get to where they need to be. Here in B, this is showing you exocytosis, and it's virtually the identical process, but it's in reverse. And so we have a vesicle that's a membrane-bound bubble with specific proteins or specific enzymes, and they will be excreted, or eventually they will travel to the membrane. They'll bond with that membrane, and they will basically be released to the atmosphere, and then those could be taken in by another cell, and so on. But endocytosis, exocytosis. Amoeba hugs are often fatal. His little buddy here would be ingested by a endocytosis event. If you ever found yourself friending an amoeba, you can still be friends, but keep your distance, because if these friends were ingested accidentally even by Mr. Amoeba here, who is completely oblivious to everything around him, your vesicle, which you would be in if you were taken in or ingested by an endosymbiosis event, you would eventually bond with a lysosome. And if you remember from our organelle discussion, lysosomes are vesicles that contain hydrolytic enzymes, or enzymes that carry digestive enzymes. And so you would eventually be engulfed by Mr. Amoeba here and would completely be digested by some of those enzymes once your vesicle and the lysosome fuse. Phagocytosis is a type of endocytosis in which it means cell eating. And so this is a process that takes in large amounts of material, and we typically talk about a large amount of solid material, which is why we say eating. Okay, and so this is a key role in your immune system. And so anytime you have bacteria, viruses, or any kind of other infection or pathogens, it's going to be taken in and destroyed by macrophages. And these macrophages are special blood cells that are going to be pros at this phagocytosis process. And so phagocytosis is just like it sounds. It's cell eating, and it's a form of endocytosis. And we're going to take in these types of materials in order to destroy them. It's one of the ways your immune system works. Phagocytosis, just when you thought it was safe to get back in the plasma. Viruses, bacteria, protozoans just swimming along, minding their own business. Then there's a big cell which is waiting with its big jaws, and those jaws are waiting to engulf unsuspecting protists and unsuspecting prokaryotes from the depths. Obviously a Jaws reference. Great movie. I couldn't swim for days. Exocytosis. This is the opposite of endocytosis. Remember, exocytosis is a form of it's a process in which we can get rid of materials. And so exocytosis is a way for the body or the cell to get rid of material. And like I showed you this before, this one was our exocytosis over here. And so if these are potentially proteins or enzymes that we want to get rid of, we're going to move them through the cytoplasm. They're going to bond with the plasma membrane. It's going to open up and it's going to release these materials to the outside of the cell. And this is a way for the cell to get rid of any kind of waste products. It's going to dump the material into the exterior of the cell, and it's just a way for the cell to get rid of things that it doesn't necessarily want. And the last picture of the lecture is a picture of the cell, and in this picture we can look at every single type of bulk transport we've talked about today. Notice that these food particles, which are over here, are coming into the cell this way, and when materials like food come into the cell, they're going to pinch in, and it's going to drag this membrane with it, and eventually this membrane is going to completely enclose it, and it's going to form this food vacuole or a vesicle, and it depends on what it's carrying, but in this case, because it's food, water, or waste, it's going to be a vacuole. If it's a protein or any kind of enzymes, it is going to be a vesicle. And so these vacuoles that are containing these foods that were brought in via endocytosis are going to be transported through the cell. Eventually, these lysosomes that are a form of bulk transport as well, or at least kind of like bulk transport, in that they are vesicles that are carrying digestive enzymes. Remember, they bud off of this Golgi apparatus and they travel to a vacuole. And once they meet, just like in that amoeba joke, they're going to actually bond, and they're going to dump these digestive enzymes into this vacuole, and that's how the digestion of this food is going to happen. The digestion is going to take place, and then eventually all of these nutrients and minerals can be absorbed by the cell. But just like any, sorry about that, but just like any digestion of any metabolism, there's going to be waste products. And just like other organisms, the cell wants to expel the waste products, and because these waste products are not needed by the cell, they're going to eventually travel to the membrane. This vesicle or vacuole is going to bond with the outside cell membrane of this cell, and it's going to actually release these materials to the side or the exterior of the cell. And so this event right here is an exocytosis. Remember, we're exiting materials, and then this over here is an endocytosis because we're taking materials in. And so one endocytosis will bring in food. The vesicle that is containing these lysosomes or digestive enzymes is going to bond with this food particles, and then eventually the waste is going to be expelled. That's exocytosis. Endocytosis, exocytosis. If this was a type of bacteria that is being taken in by the cell, then this would be a phagocytosis event. But because it's food, we just call it regular endocytosis. But that's going to be the end to Chapter 7, Lecture 9. Hope you enjoyed. See you tomorrow. Bye.